Hey class, welcome to Lab 5.2 and 5.3. Before you get started on this video voiceover, I would recommend that you read page 267 to 270 of the lab. This gives you a nice background and an introduction to the differential tests that we'll be doing over the next several weeks. And this context is very important when you're trying to put these labs together for your lab practical that's coming up next month. So in addition, I'm going to start recommending the different questions that you can do on the data sheets like we used to do in the face-to-face class. Of course, these are all just recommendations now because I can't check your data sheets. I'm just going to kind of give you a heads up in terms of what questions I find important. If there's any that I want to leave out, I'll let you know. But for these two particular labs, all the questions are relevant. All right, so let's go ahead and get started. And I want to let's remind ourselves what the point of differential tests really are. What's the goal of this? Well, the goal is to characterize the bacteria and therefore be able to ultimately identify that bacteria. And once we have that identification, we can now figure out a treatment approach. So remember that ultimately in terms of the medical microbiology field anyway, we're taking a patient sample, patient has an infection, and we're going to take a clinical sample from that patient and try to determine what bacteria is causing the problem. And once we know what bacteria it is, then we can determine a treatment approach. Bacteria have huge differences in terms of their susceptibility to different types of antibiotic and treatment approaches. And so knowing the bacteria will help us with that treatment approach. Now, up to this point, we've really done, mostly in class, what we've done is microscopy. And microscopy has a big shortcoming. This was actually a question on the practical. Of course, the big shortcoming or the big downfall of pitfall, I guess I should say, of microscopy is that most bacteria look alike. There's very subtle differences that can't be picked up necessarily just simply by looking at the bacteria under a microscope. And we can't narrow down the bacteria to a single type of bacteria when we have all these options. So think about how difficult it was, the morphological unknown, and you only had, I think, maybe like 10 different bacteria, and you knew what the results were supposed to be. So imagine having a clinical sample where you have a very small amount of bacteria. It's a wild type bacteria, so we don't know the growth requirements for this particular bacteria. You don't necessarily have a pure sample, an isolated sample, and you literally could have anything, right? You have no idea what kind of bacteria you could have. Narrowing it down simply using microscopy is very, very difficult in a clinical setting. And up to this point, up until recently, taking a genetic approach to identification was basically impossible. It was when I was an undergrad and I took micro, this wasn't even really discussed. It was kind of one of those future possibilities because trying to sequence or fingerprint bacteria based on its DNA sequence was way too expensive and way too unreliable using the technology that we had way back in the Stone Ages. And now, not only is it becoming less and less expensive, it's becoming really, really fast and very reliable. So this is definitely where the field is moving towards genetic identification of these bacteria because it's so cheap and fast. What we're going to be doing for the next month or so is looking at how we've historically identified bacteria in a clinical or in a diagnostic lab, and that's through biochemical tests. And what we're really doing is looking at the conditions that they need for growth. What does this bacteria like to eat? What are its waste products? What does it need to grow? Basically, what are its metabolic markers? And here's an example. If we were still doing a face-to-face lab, we could maybe do one of these. I try to do one of these towards the end of the semester just for fun. These are called API strips, and they are actually used in a clinical setting. And so what you do is you take the patient sample and you inoculate each of these little capsules. And you probably can't see this very well, but each of these capsules have like a little symbol, and it tells the reader what particular substrate is in these little capsules. And based on the results, based on the color that it turns, you can determine whether or not what your bacteria is. You can easily identify the bacteria, I shouldn't say easily, you could quickly identify the bacteria, and it gives you a pretty good sense of what bacteria you are looking for. So we're kind of doing API strips, but one at a time in dishes and in tubes instead of on a strip. So when we look at these differential tests, what we're really doing is looking at all the different types of metabolic activities that these bacteria can have. And there's basically 5 major categories here that we're going to be looking at. The first one that we're looking at this week are the fermentation of specific carbohydrates. And we're going to use the phenol red and the MRVP test to do that. Next week, we're going to look at the aerobic or anaerobic respiration capabilities of bacteria using doing the oxidase, catalase, and nitrate tests. It's also possible that certain bacteria can utilize a very specific component of the media for either nutrients, well, probably for a nutrient source. For example, urea and citrate tests will test for its ability, a bacteria's ability to utilize urea as a nutrient source, or citrate, which is a type of organic acid, as a nutrient source. We can also look at the bacteria's ability to utilize certain amino acids and use those amino acids as part of, basically process those amino acids and put them into their central metabolic pathways. And so a portion or a way that they can do that is to decarboxylate the amino acid, and that amino acid can then be turned into some intermediate into glycolysis or the transition step or the Krebs cycle. Finally, we're going to look at the bacteria's ability to hydrolyze macromolecules outside of the bacteria using exoenzymes. And we're going to look at the 4 major macromolecules in terms of bacteria's ability to import them into the bacteria itself. So we're going to look at carbohydrates, nucleic acids, lipids, and proteins. So these are the 5 major fields in terms of metabolic activities that we can characterize bacteria, that we can utilize to characterize bacteria. And of course, we're going to do the first one this week, the fermentation of carbohydrates. We've actually already done this. So we've already done a differential test, and that was the MacConkey agar. So I want you to just do a quick little self-check here, pause the video, and ask yourself what the substrate of this agar is, what the indicator is. How do you interpret the results of this plate? How would you characterize A, B, C, and D? And which bacteria are positive and why? How did you come up with that conclusion? So go ahead and pause the video, take a minute, and try to see how well your note-taking is from last week. All right, so let's take a look at this together. So the substrate, of course, is lactose. Remember that MacConkey agar is not only selective for gram-negative bacteria, but it's differential for, it differentiates bacteria based on the bacteria's ability to ferment lactose to acid end products. So the substrate, we can't determine this if we don't have lactose. So of course, lactose is our substrate of choice. And the indicator is a pH indicator called neutral red. So this test will differentiate between bacteria that can ferment lactose to acid end products and those that cannot. So when we look at B and D, for example, here, let me ask you, so which of these bacteria are gram-positive and gram-negative? Hopefully you recognize that, of course, all of these bacteria are gram-negative because they're growing, right? If they were gram-positive, we wouldn't even see them. We would just see like kind of a scratch that the loop made on the agar, but we certainly wouldn't see any growth. And we see growth on all four of these bacteria. Now, B and D did not turn that pH indicator red, the neutral red indicator red. So we know that B and D are negative. Even though they're gram negative, they're negative for lactose fermentation because we don't see that color accumulation. Now, C is definitely positive. We see that nice red color develop in the bacteria. Therefore, we know that whatever this bacteria is, bacteria C is able to ferment lactose to an acid end product. And we know that A can really do this, right? We see that A is such a vigorous lactose fermenter that it is precipitating the media around it. And so this is probably E. coli. E. coli does this. It's a kind of a hallmark characteristic of an E. coli bacteria where they ferment. They're such good lactose fermenters that they produce so much acid that it turns the media surrounding it pink as well. You see that nice halo around the bacteria. So you've already done your first differential test in the MacConkey agar, but let's look at the differential tests in a little bit more detail before we dive into the 3 that we are doing today. So remember that, remember this beautiful picture, this graph on page 597 that looks terribly complicated, but let's again just kind of boil this down, boil this flow chart down to its important components. There's really 3 things that this page, all of these different pathways have in common. The first one, of course, is that they all start with pyruvate. Remember that fermentative bacteria, fermentative pathway is only doing glycolysis. It's a very low energy, oxygen independent pathway. And it takes that product pyruvate and it has to somehow use pyruvate as an electron acceptor so it can recycle all of its electron carriers. In this case, it's NADH. So if its NADH can't accept any more electrons, then glycolysis can't happen and the bacteria is now in big trouble because it has 0 way of producing energy. So the second thing that all of these pathways have in common is, of course, that it has to recycle these NADHs. It has to oxidize these NADHs somehow to get rid of those hydrogens and electrons and energy. So it's going to donate those electrons to pyruvate in some way and turn it into our third common feature of this flowchart, and that is that it's going to turn pyruvate into either an acid, an alcohol, or a gas. So remember, even though these fermentation pathways are very complex, they all have 3 commonalities. It starts with pyruvate, oxidizes NADH so it can recycle it, and it ends within an acid, a gas, or an alcohol. Now, when we look at these 3 potential products, acids and gases are actually quite easy to detect in a bacterial sample. An acid, we can simply detect changes in pH. A gas, we can look for the accumulation of a gas. However, the third one, an alcohol, is pretty difficult to detect. At least it's not nearly as easy as detecting an acid or a gas. So most of these fermentation tests focus on the production of an acid or a gas. So let's look at our first test. Again, this is lab 5.2. I think it starts on page 279 of your lab manual. And this test is really many tests. It can be many different tests. So let's look at the recipe for this test and then I can explain what I mean by that. The broth itself is a very basic broth that only has a few ingredients. It has digestive casein, and the digestive casein provides amino acids for the bacteria. It has sodium chloride in it to make sure that the osmotic pressure is correct in the broth itself, and it has an indicator, pH indicator, phenol red. And then here's where all the different broths differ. So we can make, I don't know, there's at least a dozen different phenol red broths, for example, that we make in our lab. And the difference between them is the type of substrate that you're going to put in it. So you can change this, and this is what makes this phenol red test a really nice versatile test, because you can test for basically any carb that you want to add. So the ones that we typically do in class are glucose, sucrose, and lactose, but you can add anything. And you only want to add one. So you want to, if you add all 3, for example, to this tube, then you don't necessarily know which one is fermenting. So you only want to add 1 carb to your tube so that you can say, for example, if it's fermenting, if you see evidence of fermentation in this tube, you know it's specifically fermenting glucose. So in this way, we can adapt this phenol red broth to look at any carb that we choose. So we're going to add the carb to the broth itself. And then one last thing that we can't forget, even though it's not really technically part of the recipe, I want to include that we add what's called a Durham tube. And a Durham tube is like, it really looks like a tiny little test tube, and you invert it, and I forgot to put it in this one, but it should be in this test tube as well. But it's a tiny little glass tube that's open on one end, and you invert it and you put it into the media. And what this does is it traps any gas. So imagine here we have our bubble. So let's just use this one as an example, right? We have our bacteria that's kind of hanging out at the bottom of our test tube. It's growing, it's producing gas happily. And as that gas kind of travels up, right, because gas is of course lighter than the liquid, it's going to want to float. And as it floats up, some of it's going to get trapped in the Durham tube and it collects as a bubble. So after some period of time, 18, 24, 48 hours, we can see accumulation of gas as a bubble in the Durham tube. So it's a nifty little way of seeing if gas is being produced or not. All right, so let's take a look at all the possible outcomes of a test with phenol red. So here we have unknown, let's just say unknown bacteria X. We're looking at a bacteria of choice and we want to see whether it can ferment. In this case, we're going to look at glucose. So we're going to inoculate this glucose phenol red glucose broth with bacteria X. We're going to incubate it at 37 degrees for 24 hours. And we have all these various outcomes. Of course, I wanted to throw this in. I mean, again, we take this for granted, but don't forget that you could have no growth, right? We want to make sure that we don't have, we don't forget that this is a possibility. The media maybe doesn't support growth of this particular organism. We didn't inoculate correctly. Who knows but if the conditions aren't correct for this bacteria to grow, we could potentially have no growth and it should look basically like an uninoculated tube. All right, so what are our possibilities? So we could have a turbid broth. So again, imagine that this broth is cloudy, but it didn't really change color. There's no difference in the color between these two particular tubes. And in this case, we can confidently conclude that there is no acid being produced because we didn't see a change in pH. The media stays that red color. Now, if the media, let's skip down to this one. If that media were to change to either an orange or a yellow color, we know that the pH has went down and therefore an acid must have been produced. So we would say that this is positive for fermentation of glucose because we have acid production. Now, we could further characterize it's possible that we not only have an acid being produced, but we could also have a gas being produced. And we would see that gas production as that bubble in the Durham tube. So both of these are positive results for the fermentation of glucose by bacteria X. We could simply have positive for acid production, or it could be positive for both acid and gas production. And we want to make note of both of those because we can characterize bacteria based on these results. All right, so we have one last possibility, and this is an important one because it also tells us something about the bacteria, and we could have deamination occur. And if you think about, so if the bacteria can't utilize glucose, the only other thing it has to eat is this digestive casein, which remember is amino acids. And bacteria can use amino acids, just like, for example, we can eat protein and we can utilize protein as a nutrient source. Some bacteria can do this as well. And what they do here, what bacteria do, is they're going to remove the amine group, that NH3, from the amino acid. And removing that NH3 is going to cause the pH of the broth to go up. In other words, the broth is going to turn basic. And what you're going to see is this really pretty hot pink color accumulate in the media itself. So when you see bacteria go, when you see the tube, the media go from a red color to a pink color, you know that there's definitely no fermentation occurring. And what's really happening here is deamination. This bacteria is able to utilize the casein as a nutrient source, and it's deaminating those amino acids to make the pH of the broth basic. So be sure, there's a really nice table on page 281 that tells you how to interpret these results, and it helps it goes along with what I just talked about here. So let's look at a real example. And it's important to point out that even though I'm giving you these nice clear colors, right? Yet red, pink, and yellow, for example, in a lab setting, the color changes are actually really subtle. For example, would you consider this tube red or orange yellow? How would you grade this particular tube? And I'm hoping that this demonstrates the need for controls here, right? We really need to see what an uninoculated control looks like, or a negative control or a positive control looks like. And we can see that here. So let's look at these results. Let's imagine that we have bacteria, some are unknown bacteria, and we want to judge whether or not it can utilize specific carbs. So we have carb A, carb B, carb C, and carb D. tube E is going to be our uninoculated control. So this is what a negative result should look like. And so comparing tube C to tube E can see that we definitely have no production of acid. So we have negative for this particular carb C. We can see that our bacteria does not utilize this carb for fermentation. If we compare tube D to tube E, we can see that this tube looks pink compared to our control tube. And so here we can see that deamination is occurring. So not only are we not fermenting, so it's negative for fermentation, but deamination is occurring. So now let's look at our first tube, the tube that I showed in the first example at least, tube B. We can see that compared to our negative result here, C or E, that tube B clearly shows a decrease in pH. So we know that acid is being produced, and so our bacteria can ferment this particular carb, whatever, maybe it's glucose, sucrose, lactose, whatever it might be. So this is positive for acid production. While in tube A, we see that it's not only very clearly lowering the pH. We have a nice yellow result here in our media, but it's also has gas accumulation because we can see that bubble that's been trapped in the Durham tube. So we have A and B are both positive results. B would be positive for acid production. A would be positive for acid and gas production compared to our uninoculated control E. So here's how we interpret these results. Phenol red, the phenol red broth. So let's take a step back here and really think about what we're actually testing for. Let's remind ourselves how glycolysis works. So remember that glucose turns into glucose 6-phosphate. Glucose 6-phosphate turns into fructose 6-phosphate. Fructose 6-phosphate eventually turns into pyruvate, and so on, right? This is glycolysis, and the starting material for glycolysis, of course, is glucose. So whatever carb it is that we're testing for, lactose, sucrose, mannose, xylose, and so on, all the various different carbs that we can ultimately use this test to test for. What we're really testing for is if that bacteria can turn that carb into glucose. Because we can't take, right, here for example, here's lactose and sucrose, right? They're both disaccharides. I'm gonna pause the video here really quick because the garbage truck is coming by and they're really noisy. Hold on. Okay, they're gone. So let's continue. So remember, let's look at lactose here and sucrose here. These are both disaccharides, right? They have two subunits and they can't enter glycolysis directly. They have to be, they have to be converted first into glucose. So what we're really testing for, if we're looking at a carb other than glucose, what we're really testing for is that bacteria's ability to turn lactose or sucrose, for example, into glucose and ferment it. So what we're really looking for is the enzymes. Remember, enzymes are responsible for everything. So if we have a bacteria that's able to ferment lactose, what we're really doing is saying this bacteria is beta-galactose positive. It has this enzyme that can take lactose and turn it into glucose and therefore take it down that fermentation pathway. Whereas in sucrose, for example, if a bacteria is able to utilize sucrose as a fermentable carb, what we're really looking for is the enzyme sucrase. Because without this enzyme, this bacteria can't process sucrose into glucose and therefore chunk it into that pathway, that glycolytic pathway, to ultimately ferment it. So when we look at bacteria, again, other than glucose, what we're really doing is looking to see if that bacteria has the enzymes capable of processing these carbs into glucose. Let me just make sure I'm recording here. Cool. Okay, so another thing that I'm going to change moving forward here is I'm going to actually show you actual results for you to interpret. So E. coli was one of the bacteria that we would have inoculated in lab. So I'm going to do a fake inoculation for you here, and I'm going to inoculate E. coli into the following test tubes. We incubate it for 24 hours at 37 degrees Celsius, and notice that we have a base broth. So we inoculated we inoculated E. coli into a base broth that contains no carb, right? It's a nice control for us to make sure that it can't ferment in the absence of carbohydrates. And we're going to inoculate it into phenol red that contains either glucose, lactose, or sucrose. So what do each of these tubes tell you about E. coli? So interpret these results, and then there will be questions on the quiz based on your interpretation of these results. So I also want to give you a couple of challenge questions here and see if you can kind of figure out what's going on here. So we have an unknown bacteria X, and I inoculated phenol red that contains sucrose. So I inoculated this bacteria into phenol red sucrose broth, incubated 24 hours, 37 degrees Celsius. And as the results, you can see the results here. I have a bubble, which tells us that fermentation took place, but the media is pink. Let's assume that all of our controls are good. And this question really addresses sensitivity and specificity. So ask yourself, what could have happened here? I have another one for you too. So now this bacteria, same thing, same situation, sucrose broth, we inoculated with our bacteria of choice. We waited, in this case, we're going to wait 72 hours, and that's important. That's another indicator. That's another hint for you for what could have happened here. And when we get the same result, we see a pink broth, but we also see a bubble. And just a heads up, this relates to question 2 on page 284 of your data sheet. So see if you can figure this one out too. All right, let's move on to our next test. So the next test is called the MRVP test. It stands for methyl red Vosges Proskauer. And it's really important students get this kind of conflict. They fuse these two tests into one in their mind, and it's really two separate tests. One test is called the methyl red test, and the other is called the Vosges Proskauer test. And the reason that we tend to chunk them together is that we use the same broth for both of these tests. And the broth is a very, very simple broth. And we do this just for simplicity's sake so that we're only inoculating one tube. So we're saving time, we're saving space in the incubator, and so on. And so if you look at the recipe for the broth that's used in these two tests, it's a very, very simple type of broth. It has peptone and glucose for nutrient, for nutrition for the bacteria. And then it has a phosphate buffer simply to basically buffer against any small changes in acidity, or acidity or alkalinity. So remember, a buffer, basically a job of a buffer is to resist large changes in pH. So we add a little phosphate buffer in there so that the pH doesn't change too dramatically. All right, so let's look at let's treat these as two separate tests. So we have a methyl red test. And the methyl red test tests for the ability of a bacteria to ferment glucose to acid end products. And it's important to point out that these particular acid end products are very stable. They're going to last a very long time. Most acid end products that occur as a result of fermentation can either get consumed in a different pathway eventually, or they break down over time. The difference in the methyl red test is that these acid end products tend to stick around over days, potentially. I've done a methyl red test after like a week or so. So you can actually let these go for a really long time and still get a positive result because those acid end products are quite stable. So if we look, here's the pathway, right? Remember, glucose eventually will turn into pyruvate, and we need to get rid of those electrons on the electron carrier. So we're going to turn pyruvate into something else. And those something else are these stable acid end products. For example, we could have lactate, which turns into lactic acid, succinate, which turns into succinic acid, formate, which turns into formic acid, and finally acetate, which turns into acetic acid. Notice that we also can have a little bit of alcohol production as well, but this test is not detecting ethanol. It's only detecting these molecules that will decrease the pH. So in this particular case, what is the substrate? What is the indicator? Right? Ask yourself, remember, every differential test guaranteed questions on the practical that will test your ability to understand how the substrate and indicators work in each particular test. One of the things that's different about the methyl red test in comparison to, say, oh no, they're back. Hold on, let me pause it. Okay, sorry about that. The joys of quarantine, of this big bay window that's right outside of where everyone positions their garbage cans for pickup, and it's so noisy when I'm usually at school when this happens, so I'm not used to this. So anyway, okay, so we were talking, where were we? Goodness. Oh, so the difference between the methyl red test and let's say the phenol red test or the McConkey test is that here the indicator isn't actually part of the broth or the agar itself. We're going to add it after. So we're actually going to incubate this first for 24 hours, 48 hours, whatever it might be. And then we're going to add a drop of the methyl red indicator. Again, just a pH indicator, methyl red. And we're going to add it after to get our result. And so the only positive result that we should see is a red result, because that's going to indicate that our pH is low. Anything other than red, it usually turns this like kind of yellow coppery color. That's a negative result. So after we add that methyl red, within 5 seconds, because remember, this is just a pH change, pH indicators will change right away. This is going to tell us that we have some type of stable acid that's being produced in this particular pathway. Now, the Voge-Proskauer test, on the other hand, are also able to ferment, but they have a different pathway and a different end product. So let's look at that first. Remember, we have glucose, of course, turning into ultimately pyruvate, but you can see that bacteria that are positive for Voge-Proskauer are going to do something different with these, utilize this pyruvate in a different pathway. It can produce ethanol as well. It can produce lactate and formate also, but if you notice this main pathway here that it takes, it can turn into, ultimately, it will turn this pyruvate into a product called 2,3-butane-diol. And this is what we're looking for. Ultimately, we're looking for the bacteria's ability to produce 2,3-butane-diol as an end product. And so we do see a slight initial, we see fermentation lead to a slight decrease in pH initially, but that will stabilize itself very quickly as these acid end products go away and we have 2,3-butane-diol accumulation in the media. So the bacteria kind of sense this fermentation pathway and they switch to this 2,3-butane-diol pathway. And again, similar to the methyl red test, we're going to use the indicators after incubation, and we have two of them. In this case, they're called Voge-Proskauer A, Voge-Proskauer B. And these reagents test for an intermediate in this pathway called acetoin. So when we add VPA and VPB, it's going to take acetoin. These two reagents are going to react with the acetoin, turn it into diacetyl. This diacetyl reacts with guanidine that is present in I think the guanidine is in the peptone, and that's going to cause a red color to develop. So this is very different if you think, and it's very different than what we've done up to this point. Think about the phenol red, McConkey, MRVP, or the methyl red test. What we're doing here is simply looking for a change in pH. This is actually a chemical reaction, right? These reagents are going to cause acetoin to turn into diacetyl. Diacetyl reacts with guanidine, and a red color develops after. We actually have to wait, I think it's like up to 30 minutes. It can take 30 minutes for this reaction to take place. And we also have to shake it periodically. We have to kind of agitate the tube that we're reacting this broth in because it needs oxygen to happen as well. So this is a very different test in its approach because it's not simply using a very simple pH indication to give us a positive result. We actually have some chemistry going on here in the detection of these intermediates in this 2,3-butane-diol pathway. So if we look at these two tests, here's what we would have done in class. We can take two different bacteria, E. coli and Escherichia coli, and I think this is Enterobacter aerogenes. They were inoculated into the MRVP broth. They were incubated for a certain period of time. And then you separate the broth tube so that you can do your two different tests, the methyl red and the Voge-Proskauer test, and you can see the results here. So when we add methyl red to E. coli and E. aerogenes, we get a red color develop right away in the E. coli, but the E. aerogenes will stay that yellow color. Whereas when we add the Voge-Proskauer reagents A and B, we wait 30 minutes, we agitate the tubes. E. coli remains yellow, whereas Enterobacter aerogenes is going to turn red. So interpret these results. You can write them down in your data sheet as if you did them, right? And use these to address the questions in your data sheet. So let's just take a minute here and compare and contrast. These will be a lot. There'll be a lot of questions on your second lab practical that will compare two different tests. For example, I might ask what the differences, the differences between the methyl red test and the Voges-Proskauer test as, why you would use one as compared to another. So these two tests, of course, have similarities and differences. Of course, the similarities, they use the same broth. They're both characterizing bacteria based on fermentation products. We're adding the indicator after the bacteria is incubated, whereas in the previous phenol red or MacConkey, the indicator is actually part of the broth itself. And not that it's terribly important, but they both have a red color as an indicator of a positive test result. But they also have really important differences. Of course, they're looking for different fermentation end products. I forgot to even add that in the difference. Of course, the most important difference is that they're testing for different things, right? In methyl red, we're testing for stable acid end products from glucose fermentation, whereas in Voges-Proskauer, we're specifically looking for the production of 2,3-butane-diol. Now, after we add our indicator, another difference, of course, is that after we add our indicator, we should see an immediate result in the methyl red because we're simply looking for a change in pH, whereas the Voges-Proskauer test can take up to 30 minutes to give us a positive result. Also, let's think about how acid works in both of these tests. In the methyl red test, we're going to see a very clear and sustained drop. In other words, a turn towards an acid pH in the methyl red because of those stable acid end products. Whereas in the Voges-Proskauer test, we only see a small drop in pH, and it's not going to last very long either. As that 2,3-butane-diol, which is a neutral end product, as that accumulates in the media, we're not going to see that pH stay low for very long. It kind of stabilizes quite quickly, and therefore we can't look for a change in pH based on the development of this 2,3-butane-diol. So it's also important to point out here that first of all, both of these tests are part of a very important set of tests called the IMVIC test. So IMVIC stands for indole methyl red Voges-Proskauer citrate. So there are indole methyl red four four tests again. And I'll talk about this again in later labs. Indole, methyl red, Voges-Proskauer, and citrate. So these four tests, between these four tests, it will distinguish between members of the enteric. So the Enterobacteriaceae family, they have a lot of common pathogens in them. And so it's very important to utilize tests that can differentiate between these different, the different genuses within this family. So for example, Escherichia, Shigella, Salmonella species are all methyl red positive, whereas Enterobacter, Serratia, and Erwinia species are Voges-Proskauer positive. So you might ask, you're probably in the, when I was talking about the Voges-Proskauer test, for example, you're like, who cares about 2,3-butane-diol? Like, what is this? Why does this even matter? Like, what's the point of this, right? How did we even come up with this, right? Well, it matters because it's a characteristic of metabolism for some very important pathogens, the Enterobacters, this S. maricens. I don't know if we worked with this one. It's a really cool bacteria. It turns a really pretty red color. But so these three families, Enterobacter, Serratia, and Erwinia, all produce a hallmark characteristic of their metabolism is that they produce that 2,3-butane-diol. That's why it's important, because we can use it as a characteristic of metabolism for species that fall into these three families. Okay, I think that's the last. Yeah, that was the last slide for this particular lab in less than 40 minutes. That's not too bad. Hope this is working for you guys. I'm going to put up a survey here actually this week to ask for some feedback on how these voiceovers are going and anything else that I can do for you guys in terms of helping you along with the class. So I hope you're enjoying it so far.